Hello, I'm Binoy. Today I will talk a bit about our research towards developing new methods for single cell and single molecule screening. The development of analytical tools for early stage diagnosis of diseases can often be limited by low abundance of particular biomarkers. This is compounded by the complexity of probing analytes within biological fluids. Single molecule detection methods, on the other hand, enables rare event detection in complex biological fluids without the clouding associated with ensembling averaging for accurate detection and quantification of ultra-low concentration biomarkers. At the EDEL research group where I work, we employ different single molecule screening strategies for detection and manipulation of individual molecules with high spatial and temporal resolution. Today in my talk, I will focus on our work in developing a new tool for selective trapping and manipulation of single entities from individual cells. The grand challenge in any single molecule detection strategy is to confine the analyte molecule within a small detection volume. Unfortunately, the dominant mechanism that enables the confinement of analyte molecule in this detection volume are diffusion limited, resulting in only a small fraction of the total sample volume being accessed. For example, a 10 microliter solution at a concentration of 1 nanomolar will have an average of 34 molecules in the capture volume, assuming a capture radius of 3 micrometer. This is roughly 10 to the power of 8 times lower than the actual number of molecules in the solution, leading to long measurement time scales. So we need new strategies to enable rare event and needle in a haystack kind of detection experiments. In our research, we overcome this limitation by incorporating a dielectrophoretic trap to the single molecule sensor for selective confinement of the analyte molecule from ultra-low concentrations. Dielectrophoresis occurs when a polarizable particle is suspended in a non-uniform electric field. The electric field polarizes the particle, and the poles then experience a force acting on the field lines, which can be either attractive or repulsive according to the orientation of the dipole. Since the field is non-uniform, the pole experiencing the greatest electric field will dominate over the other, and the particle will move. The orientation of particle is dependent on the relative polarizability of the particle and the medium in accordance with Maxwell-Wagner polarization model. Since the direction of the force is dependent on the field gradient rather than the field direction, dP will occur in AC as well as DC electric field. The polarization and hence the direction of the force will depend on the relative polarizabilities of the particle and the medium. If the particle moves in the direction of increasing electric field, the behavior is referred to as positive DEP, whereas if acting to move the particle away from the high region, then it is negative DEP. By carefully engineering the electric field, we can make the analyte molecule to move towards the sensing volume. This is particularly powerful technique, especially for the detection of molecular species present in low copy numbers, which are currently elusive to the state-of-the-art methods. To trap and concentrate nanoscale entities such as biomolecules, dielectrophoretic forces in the order of femtonewtons are required to overcome Brownian motion, convective flow due to heating, and electrohydrodynamic effects. As the field strength is proportional to the applied voltage and the distance between the electrodes, larger trapping forces can be achieved either by increasing the voltage between the electrodes or by reducing the distance between them. However, application of a higher voltage can lead to unwanted heat generation, bubble formation, and electrochemical reactions, and hence is not desirable for manipulating biomolecules inside or outside living cells. To overcome these challenges, we developed a nanoscale dielectrophoretic tweezer with a very small electrode separation in the order of few tens of nanometers. We fabricated these nanotweezers using nanopipettes made from double-barrel quartz capillaries via a laser pulling. Nanoelectrodes were fabricated at the electrode tip by using pyrolytic deposition of carbon. As you can see the SEM and TM images, the nanotweezers were consist of two coplanar semi-elliptical electrodes of approximately 20 nanometer in diameter. The two electrodes were separated by a quad septum of 10 to 20 nanometer in width. Elemental analysis of the fabricated nanotweezer confirmed the presence of a continuous carbon deposit inside the nanopipette. The close spacing of two coplanar carbon electrodes offers the possibility of generating high dielectrophoretic forces without employing high voltages. From the finite element method calculations that we performed, field gradients as high as 10 to the power of 28 volts square per meter cube near the electrode gap could be observed. With such high field gradients, single DNA molecules well below 200 base pairs could be trapped. The z-axis projection of the field gradient along the tip can be seen here, where a highly localized trapping field can be seen at the very end of the tip. It is possible to operate the nanotweezer at voltages as low as 1 volt peak to peak to minimize the effect associated with the heating, especially in higher conductivity solutions such as cytoplasms. The effectiveness of the nanotweezer was experimentally validated by trapping and extracting fluorescently labeled DNA of different sizes and small proteins such as monomeric alpha-synuclein. Here you can see the results of trapping of 10 kbp DNA molecule from its solution. The application of an AC voltage at a frequency of 1 MHz and a voltage of 20 volt peak to peak resulted in the accumulation and concentration of DNA molecules at the nanotweezer tip. Leveling of efflorescence intensity was observed soon after the AC field was turned on and attributed to the saturation of DNA accumulated at the tip. When the AC voltage was turned off, the fluorescent intensity decreased as the molecule diffused away from the tip. You can see a video of this trapping and release here. So in the solution, you can see fluorescently tagged 10 kbp DNA molecule. On application of a DEP field, you can see the DNA start accumulating at the tip. And as long as the DEP is applied, the molecule stays there. But once you switch off, the molecule diffuses away. So this process can be repeated. In the same way, we can trap alpha-synuclein monomers. Here you can see the solution contain fluorescently labeled alpha-synuclein monomer that will start accumulating at the tweezer tip when we turn off the dielectrophoretic field, and they diffuses away when we switch off the dielectrophoretic field. By decreasing the DNA concentration down to 100 femtomolar, it was possible to optimize the nanotweezer to selectively trap and extract single molecules for further analysis. Here you can see fluorescent intensity versus time traces recorded for two tips in which a single molecule is trapped and another tip in which 3 molecules are subsequently trapped. The corresponding qPCR amplification curve showed 100% amplification of the trapped molecules. So this was performed by withdrawing the nanotweezer tip containing trapped DNA molecule from the solution and snapping it off to a qPCR tube containing the qPCR mix and amplifying it at the right conditions. Further, the sequencing of the amplified DNA confirmed an amplification with a 99% identical amplicon as the corresponding lambda DNA segment. A unique feature of the nanotweezer is that it can be used in combination with an XYZ positioning platform to perform pick and place type measurements in which single molecules can be trapped, moved at a velocity as high as 30 micrometers per second, and then released. Here you can see a typical example where a fluorescently tagged DNA molecule is being captured by the tweezer, moved, and then released. So you can do pick and place kind of experiments by using this tweezer. So you can manipulate them in different patterns. As you can see here, you can trap a molecule, then you can move it along while holding it at the tweezer tip. Having established the capability of trapping and releasing single molecules in solution, we then use the nanotweezer to perform highly localized single-cell biopsies. The biopsy method does not aspirate cytoplasmic fluids and allows for the reconcentration of analytes in real time. This is a particularly powerful technique, especially for the detection of molecular species present in low copy number, which are currently elusive to state-of-the-art methods. Also, the footprint of this tweezer is small enough not to create any damage to the cell membrane, so you can keep the cell intact while performing the biopsy. In particular, we explore the possibility of trapping and extracting DNA, RNA, protein, and organelles from a single cell. Here, to visualize the extraction of DNA, U2OS cells were stained using a DNA binding dye, and the nanotweezer tip was then inserted into the cell nucleus, and an AC voltage was applied to trap the DNA molecule. Here you can see upon application of an AC dielectrophoretic field, the DNA accumulates at the nanotweezer tip, and then you can withdraw the tip from the nucleus, and you can still see the fluorescent spot on the tip, confirming that the trapped DNA is still there, and you can extract it out to the cell, and then you can perform further analysis such as qPCR amplification. Here you can see the extracted sample when subjected to a qPCR amplification of a target sequence in 45S ribosomal DNA using a pair of specific primers that confirm the presence of 45S ribosomal DNA sequence in the extracted volume. The ability to operate at the single molecule level was then demonstrated by extracting individual messenger RNA molecules from the cytoplasm of an individual cells. Here in this case, proto-oncogene 1 transcription factor ETS1 mRNA was first fluorescently labeled with in situ hybridization, and then the nanotweezer was positioned adjacent to a fluorescently tagged RNA molecule, which was subsequently trapped and withdrawn from the original position in the cell. Similarly, we performed protein extraction. Here you can see a neonatal cardiomyocyte cell expressing GFP protein as a fluorescent part. So here we have a nanotweezer inserted into the cell. So you can see a bit of fluorescence there. But when we turn on the DEP, the fluorescence at this location increases, which means we are trapping and concentrating the protein molecules there. Now you can withdraw the tweezer from the cell, and then you can still observe at the tip of the nanotweezer. Here you can see the tip of the tweezer containing a fluorescent spot, which is the trapped GFP molecule. Finally, the nanotweezer were used to extract subcellular structures such as organelles. Here we demonstrated this by extracting mitochondria from mouse hippocampal neurons. So here it's an axon of mouse hippocampal neuron. You can place a nanotweezer close to the mitochondria, turn on the dielectrophoretic field to trap the mitochondria and then extract. Here you can see a video. So here the tweezer is positioned towards a mitochondria, and then you turn on the dielectrophoretic field, and here you can see so here trapping and then extraction of the mitochondria, and the extracted mitochondria is still on the tip. So you can see it here, the extracted mitochondria at the tip of the tweezer. So you can use this for further analysis. So having developed this, at the moment we are focused on extracting nucleic acids and organelles from the different types of cells and doing analysis to see dynamic changes in nucleic acid distribution at different locations of the cell, as well as variations in nucleic acid profiles of different organelles within the cell. With that, I would like to thank all the group members and collaborators who contributed towards the work, and the funding agencies for supporting our research. Thank you.
Hello, everyone, and thank you so much for attending this seminar and give me a chance to show you some of the projects that we are working on. My name is Ismael Diez. Everyone knows me as Isma. I'm in the chemistry department, King's College London, and we are the group of biomolecular electronics. In our group, we are interested mainly on how charged electrons diffuse across biological structures. So starting with a little bit of context here on why it's important to study electron or charge movement across these biological structures. So you will appreciate, especially those more connected to biology, that in life, electron transfer or transport is a ubiquitous process. So it's in any process that you can think of in a function in a cell involves one or multiple steps, physical-chemical steps of electron transfer or transport. Right? So here are some prominent examples that you know very well, and if you look in detail, there is a molecular machinery inside each of those process that has been evolved by nature during millions of years to translocate the charge very quickly. So nature can put charge in very effectively in any point that is required, because for the function of the cell, this is a major requirement. So the electron has to get there quickly when it's needed. And also what is very distinct in electron transport in biology, right, is that can happen in a long range. So this charge can travel long distances, right, across these chemical structures very efficiently. To put an illustrative example of this long-range electron transfer or transport in biology, here's the example of this Salmonella bacteria that create this very long pili, which the bacteria uses to translocate electrons as part of their respiration. If you look at this pili, so the building blocks are these multi-heme proteins that conform the length of this pili. And now, very recent reports of this pili, or the electrical characterization of this pili, show that the electron transfer across them is in the centimeter scale length of distances. So it can translocate actually a very efficient electrons in a very long distances. Right? Another report here in Cell, for example, 2019 as well, they revealed the crystal structure of this pili as this stack, organization of heme groups, and they also record a micrometer scale transport on this particular system. So this, although the information on the crystal structure of this pili is now very recently in the last 2-3 years well known, right? The mechanism for why the electrons can be translocated, can move such long distances between micrometers to even centimeter scale is still really unknown. So let me show you how we tackle into that problem. So our group comes from this field called single molecular electronics, where we have the ability to create electrical contacts in single molecules between two electrons in a nanoscale gap, like the one represented here. Now, with this kind of single molecule devices, we were able to look at how electron transport in chemistry is affected by things very important in chemistry, such as conformation, substitution, length, conjugation, and along, etc., etc. So this field has been evolving in the last 20-30 years, and now a group, we went one step further and trying to look at a higher level of complexity in these chemical systems and look at how biomolecules with a more complex structure perform as electric conductors, and look at more complex concepts such as chirality or the folding structure of these proteins, or the supramolecular interactions affects the way the electrons moves inside these structures. So this is a fantastic fundamental platform that we can use to understand mechanisms of electron movement in living organisms, but also we can also address all the more technical problems, such as this coupling problem in bioelectronics. So in bioelectronics, we have these fantastic sensors where we have a protein biomolecule that is the active component that will specifically connect or interact with an analyte, and then the signal is transduced electrically by putting an electrode that hybridizes to this protein underneath. Now, the way we hybridize this protein to the electron is still a question mark, and that is a bottleneck to really optimize this biological bioelectronic sensors. Right? If we think that with this platform we can really address this problem and then answer questions that are important in this kind of technologies, such as how maximize the electron-protein coupling or communication, where to put these electrical plugs in these particular structures, or how these electrified interfaces are behaving under force fields. Now, the way to trap a single biomolecule or a single protein in these nanoscale gaps is, in our case, we used an electrochemical scanning proof microscopy setup. Right? This is the case of an electrochemical scanning tunneling microscopy, where we have this STN tip and a surface, right? And we can create precisely a very small nanoscale gap between the two electrodes. This is all immersed in an electrolyte in physiological conditions with a certain pH, ionic strength for these proteins to live, to survive, to fall properly. And we have a very precise electrochemical potential difference between the two electrodes. So we can create an electrochemical potential gradient very precisely between these two electrons with a bipotentiostat, just as in any electrochemical biological setup, where you have two redox partners with two particular, with a difference in the electrochemical potential between them to exchange electrons, right? So we have been using this technique for a while now, for many years now, pioneering this particular approach. Now, when you trap a protein, right, what do you see? Right? You know, how do you detect the moment where a protein is trapped? It's connected electrically with these two electrons in these nanoscale gaps. So this is a real traces of the detection of a protein trapping process. And what you see essentially is that you're monitoring the current versus time, and then you see this kind of like jumps that we call blinks in the jargon of this field. When a blinks occurs, a protein gets trapped in between the two electrodes, and then you can measure a current level. Now, because you're measuring this in ambient conditions at room temperature in an electrolyte, right, the lifetime of these blinks are really small. We're talking about here between several tens to few hundreds of milliseconds. But our electronics are fast enough, right, for so that in this time frame, very short time frame, we can just ramp any electrical characterization that we want from the single protein junction, such as we can collect current versus voltage characteristics, transistor behavior, current versus force, spin injection, and pretty much any electrical characterization that we want to take from the single protein device. Now, the way initially to present, you know, this data, any data, is in the form of statistics, as any other biophysical single molecule biophysical approach. You have to collect lots of different traces and then construct two-dimensional representations. In this case, this is a two-dimensional blinking map that we call, or a two-dimensional conductance map, where we have on the Y scale the conductance of this single protein device versus the lifetime of the junction, right, of the protein in there. And so this kind of like lines or stripes with high content here will determine the average conductance level of our particular single protein device. Now, the process to make a connection to a single protein, redox protein, for example, in this case, we have this, I'm going to show you an example with this very simple copper azurin. This is a protein that we've been using as a test bench for the sake of simplicity. It's a globular redox protein with a copper center that gives the redox properties to this protein. And this is from a bacteria, so it's a globular soluble protein that has two natural cysteines with sulfur atoms that can be used as anchoring points on any metal surface. Now the advantage of an electrochemical STM or an STM in general is that we can, because those proteins are redox proteins, are conductive, right? We can image them individually on a surface. This is like different images. This is a big scan, and those are like two different zooming details of a Go111 surface, atomically flat surface, where we absorb several of these globular copper azurin proteins, and we can see perfectly identify single proteins on the surface. Now we can go with our tip, localize, put our tip, park our tip on top of a group of proteins, and then wait for them to stochastically form a single protein junction between the two electrodes in the way that I showed you in the previous slide. Now, when we start in this field, we were quite I would say alone, so we didn't have much data to look upon. So we start this field of single protein junctions by asking very fundamental questions. The first, I would say, logical question or obvious question to make is: are proteins good conductors? Are these redox proteins a good conductors of electricity? And before attempting that question, we have to actually, first of all, we had to make sure that these trapping events that are we identify in our gaps in the presence of proteins are due to a protein that preserves the secondary structure. It's an active protein, electrically active protein. And so among all the different control experiments that we have to do, one of the obvious is by the nurturing the protein. So we can always denature the protein by putting temperature or using different chemical agents. If the folding structure is completely broken in this protein, we see that in our we see actually a very silent gap. So we have no signal whatsoever of any trapping feature or event between the two electrons. So it just appears as if the gap is completely open all the time. Now this is opposed to when the protein is active and is folded, right? Then we see clearly these kind of features, right? I can show you many other different experiments that we had to do to make sure that we were sure that this signature, electrical signature, due to the single protein trapping. But in the interest of time, I'm going to show you a very visual picture here. This is a collaboration with our friends in Madrid. There's a theoretical group that have done a full atomistic molecular dynamic simulation of our protein junction. You see here a full protein junction, all atom simulation, even including water in this particular junction. And you see the two electrodes getting closer and closer, the tip on top and the surface at the bottom, and there's the copper arsenic ring in between. And you can see that even by squeezing the protein and with gap separations as small as 2 nanometers, which are even lower than what we're using in the experiments, right? The secondary structure of the protein is very well preserved, right? Now, if we take this conductance level signatures that we get in our single protein trapping events, right? And then we extract a value for the conductivity of the conductance of the single protein junction, right? This is the numbers that we get. Usually in this field, all the conductance values are expressed versus this G naught, this quantum fundamental constant. This is a G naught. This is the conductivity of the two electrons connected by one single atom, which represents the ballistic case where there's no scattering of electrons, so it's full transmission of this I would say atomically precise junction constriction. And now any molecular junction will be orders of magnitude below this G naught ballistic level, right? Now that number may not say much to you, but if I compare this conductance value with the conductance values using the same approach of a very simple synthetic backbone like this octane alkane dithiol, right, we see that the conductance are in the same level or even smaller for this particular synthetic backbone, which is like 3 to 4 times smaller in size than the copper arsenic protein here. So which tells us that actually these proteins are very efficient conductors, right? And actually the straight comparison between these two elements, the natural protein and the synthetic backbone, the simple synthetic backbone, tell us that the efficient conductivity observed for the protein has to be inherently in the supramolecular chemistry inside this protein. So this folding structure of the protein, the way it folds, the internal interactions, right, will define efficient pathways for the electrons to go and be efficiently transported across this particular supramolecular structures. And that initially, when we saw this, we were very excited to see these results, and we coined these effects to supramolecular electronics, which we believe is inherently in the mechanism of electron transport in biology. Following to previous question, right? Second obvious question is, what is the role of the metal cofactor on this particular redox proteins? All redox proteins has somehow a metal center that provides the redox activity to the particular protein. Now, in the case of copper arsenic, we have this copper atom in the center. And then now what we observe, if we study in detail how much is the current, the conductance of this protein junction as a function of this electrochemical potential gradient between the two electrodes of our nanoscale gap, what we observe, something very interesting, is that when we set the gradient exactly at the redox potential of this copper center, right, the current flow across this particular protein, right? So the current that is flowing in this direction of the protein, right, is maximized. So the rate for electron transport in this particular potential or gradient between the two electrodes, when it matches the redox point of this copper, is maximum. Right? This might be a small change from here to here, right? But this is an analog scale. So now if we look at the graph on the right-hand side, we see that the enhancement of the current actually is quite significant. It's about an order of magnitude increase when we tune this electrochemical potential gradient with the copper redox center of the protein. So yes, the copper redox center is actually enhancing, at least if not the key ingredient, it's actually enhancing the conductivity of the protein, and it has a clear electrical role in the electron transport across these particular protein structures. Right? Now, if we remove the copper, which is something we can do chemically very easily, right? We say actually that this enhancement disappears, and then we have this flat line of the conductance as a function of the electrochemical potential gradient of the junction. Now, from these two pictures, we can actually extract a very interesting mechanistic information. So this kind of bell shape in this particular determinations of conductance versus electrochemical potential gradient, right, is characteristic of a mechanism called two-step sequential tunneling with electron, right? Obviously, it goes into the copper center and there's escape to go to the second electrode over here in the junction, right? Now, from this formulation, we can actually extract a value that is important, which is called reorganization energy. This is the energy of the protein that needs to reaccommodate the electron. And actually, we compare these values to values for other redox synthetic molecules, right? These values are pretty low, which is a signature also of this electron transport in redox proteins, that they use and minimize this reorganization energy to translocate the electron even faster, right, in this particular setup. Now, for a non-copper redox protein, where the protein has no copper, right, we have this no-enhanced, which is characterized by an off-resonant tunneling. So the electron just goes through the protein. And this has a particular interesting point in here, is that even though the copper redox center is not there, right, the level of conductance, via this off-resonant tunneling actually pretty high. So they are about 10 to minus 5 G naught, and I show you what was the conductance for a very simple alkane dithiol synthetic backbone. So it means that copper center, although influenced, right, and is connected somehow to the electron transport through proteins, is not the only important ingredient. In the conductivity or the conductance of this protein without the copper center is already very high by itself. Right? So again, that points toward the importance of these supramolecular electronics inside these redox protein systems. Now, the previous results take us to another question, right? Obvious question, which is are there electron pathways inside these proteins? So essentially, are like in this three-dimensional folding structure, do we have specific pathways, ways for the electrons to go through the protein? Right? In order to answer this question, it's an entirely different project that we're developing, where we're trying to combine the single protein junctions with mutagenesis in order to bioengineer the single protein contact. So essentially, what we're trying to do here is we exchange some of the amino acids on the surface of the protein, and we substitute those amino acids by extra cysteines that will be the new anchoring points of the proteins to the electrode. And in this way, we will force very particular orientations of the protein in the junctions. Therefore, we'll be able to, if we repeat this at different points on the surface of the protein, we will be able to map out all these electron pathways inside the protein structure. Now, this is a complete story that I will need another talk to talk about this. But just to make the long story short, these are some of the just a summary of the results that we got and some of the modifications. So we have the wild type on the left, where we saw this bell shape of the conductance versus electrochemical potential, the characteristic of this two-step sequential tunneling. Now, these are three different modifications of surface cysteines on the surface of this protein, and these different points, amino acids on the surface. And what we observe actually is very interestingly, is that if the modification is done away from the copper center, right, like in this case of the aspartate 76 in this position, right, we actually recover. We observe equally this two-step sequential tunneling, which is the mechanism per se, right? But when the modification is done close to the copper center, in this north part of the protein where the copper is closer to the surface, right? These two sequential tunneling disappears, and then we go back to the coherent tunneling that we saw in the case that we remove the copper completely. And actually, that points to an important, plausible mechanism, biological mechanism of electron transport here that tell us that if the copper protein, right, interacts in the area close to the copper center, actually this north pole of the protein corresponds to the typical docking area of this copper protein to the redox panel, and that actually these interactions will trigger internal changes, structural changes that actually change the way the electron is translocated across this particular protein, so change the electrical properties of the proteins inherently, right? Now, we still need a lot of work to identify what structural changes, right, are producing these changes in pathways inside the protein, right? So there's a big question mark here, but I think this, I would like to at least show you a flavor of this very exciting research that we're doing in this particular directions on electron pathways in proteins. Okay, I'm going to end my talk here. I hope you get the flavor of this fascinating field of electron transport in biology. Just giving some acknowledgement here, and especially to the ERC program for giving us substantial funds to continue with this single protein junction business. And all of you, thank you so much for your attention. Thank you.
Good afternoon, everyone. Thank you for coming out to today's seminar. Thanks to the organizers for the opportunity to present my work here today. My name is Taylor Stock, and I'm a research fellow in the Scanning Tunneling Microscopy Fabrication Group at the LCN. And today I'm going to tell you about silicon surface chemistry of atomically precise arsenic doping. So I'm going to tell you about an STM-based fabrication technique that the inspiration can be traced back to two papers from 1998. The first is the famous Bruce Kane proposal for a silicon-based quantum computer. And in this design, it's required to place individual phosphorus atoms at precise positions within a silicon crystal. And they specifically need to be about 20 nanometers apart from one another and 20 nanometers below the surface. Now, this is a very tall order for fabrication in 1998 and also in 2021. But the same year, a somewhat lesser known paper came out from Tucker and Shen, which proposed a method by which one might actually go about doing this type of precise doping in silicon. And the proposal sought to exploit the recently discovered ability to desorb hydrogen from a silicon surface using an STM tip. And the proposal was that you could use this technique to fabricate a hydrogen mask on the silicon surface, which would then let you selectively react dopant precursor molecules with the silicon, providing precision doping. You would then overgrow this with silicon and repeat, and potentially build a structure like what Kane had proposed. So the group at University of New South Wales at the Centre for Quantum Computing began actually actualizing this fabrication technique and succeeded in doing so. And over the last decade or so, they've built a number of increasingly sophisticated devices using phosphorus and silicon. Most recently in 2019, they demonstrated a 2-qubit gate using these phosphorus-based spin qubits. So the technique works as follows. You start with an atomically clean silicon surface in an ultra-high vacuum, expose it to atomic hydrogen to produce a single atom thick passivating layer. This can then be patterned using the tunneling electrons from the STM tip. And now you can react your precursor gas. In the established version of the technique, this is using phosphine, which will selectively react with the exposed silicon regions through the hydrogen resist. Now, if you heat the sample up, you can incorporate the phosphorus atoms delivered by your precursor molecule into the top of the silicon lattice. And then using low temperature silicon epitaxy, you can encapsulate this in the silicon crystal. Now it's possible to remove this from the ultra vacuum chamber, and you can use standard microfabrication techniques to establish electrical contacts with your buried dopant structures. Here's an example of the hydrogen lithography stage of a quantum dot single electron transistor with in-plane side gates that we've fabricated at the LCN. Here we zoom in, take a close look at the quantum dot at the precursor exposure stage, and you can see the molecules absorbed just in the patterned region. Now, this is up to now been done using only phosphine and phosphorus, but if we can find alternative precursor molecules that are compatible with the process, we could do this using alternative dopants such as arsenic. Once we have two dopant species, you can imagine we can create increasingly sophisticated structures using the two dopant species. And once we have established a second dopant species, then maybe it's possible to introduce even more different dopants. And this is the focus of my work. And what I'm showing you here is basically expanding the materials palette of this ultra-precise fabrication technique. So why might we want more than just phosphorus available to this technique? Well, the first reason is it will allow for increased device functionality. Alongside the Kane proposal for silicon-based quantum information devices, there exist others, such as the Stoneham, Fisher, and Greenland optical quantum gate, which requires more than one dopant species. In the case of the SFG scheme, it requires two different dopants. There's also intrinsic properties of the dopants themselves, such as the higher nuclear spin of arsenic, which will provide possibilities for other advanced functionalities in the devices. On the other side of things, the materials properties themselves of the dopants and the precursors might allow for advanced fabrication procedures. For example, arsenic compared to phosphorus has a higher solid solubility and a lower diffusivity in silicon, and this might be exploited for different fabrication steps when used alone or together. So basically, we have a variety of properties of these two dopants and their precursors that will allow us to develop advanced device functionality and advanced fabrication strategies. So when we're introducing any new dopant, it turns out that there is 6 criteria that must be met by the precursor and or the dopant itself. And I'm going to show you that arsine and arsenic meet all of these criteria. We'll focus on the first 4 in particular, which require an understanding of the few and single molecule surface chemistry. So to start with, criteria 1 is that the molecule must dissociate on the surface to provide our dopant atom. Looking back to the phosphine system, much of the initial work was understanding this surface chemistry. And it turns out when you expose clean silicon to phosphine, you end up with imaging a variety of features with the STM. And these can be explained as the different dissociation products of the phosphine molecules. It dissociates on the order of minutes. And essentially, it lands at one silicon dimer site and then shuttles down the dimer row, successively dropping off one hydrogen after another until it occupies 3 silicon dimers or 6 silicon sites. And this is the preferred and stable configuration at room temperature. In the case of arsine, it's a different story. Somewhat surprisingly, you do not find this multi-species of the dissociation products. Instead, you find a single feature, predominantly a single feature, which has a notably different appearance. And using DFT and STM measurements, we find this feature, which is shown here in filled and empty states STM images, is in fact the interrow end bridge structure. That is, the arsenic is bonded across 2 dimer ends, and the hydrogens are dropped off on the immediate sites, both within the immediate dimer row and the neighboring dimer row. So this preferred structure can be explained by the following dissociation pathway. It turns out this is a kinetically preferred reaction for the arsine. And the difference is that the activation barriers for diffusion of the ASHX moieties is much higher than for the PHX. And so in fact, the hydrogen shift is the preferred reaction. And this is why the arsenic atom does not move from its initial absorption site and ends up surrounded by the hydrogen that it drops off at the closest available sites. This reaction is also happening much faster for arsine than it is for phosphine, whereas the phosphine is taking minutes to fully dissociate. The rate limiting step in the case of the arsenic is only 10 seconds. This is why when you image this thing after dosing, you see essentially the one feature. Okay, so now we know that the arsine can deliver our arsenic atoms. The next step is we want to be able to substitutionally incorporate this into the lattice. So here we start with a low density arsine coverage on the silicon surface, and here's our inter-row end bridge dissociation structures. Now, if we heat this to 350 degrees Celsius, what we end up with are these telltale add silicon dimer rows. And what these are ejected silicon that is removed when the arsenic replaces it in the lattice, and they're mobile on the surface and collect in these add dimer islands. Now, this is an indirect measure of the absorption, the incorporation of the arsenic. But if we heat it up to 350 to 500 degrees Celsius, the add silicon will actually diffuse to nearby step edges, and what we reveal is the clean silicon surface now speckled with these arsenic silicon heterodimers. These have a very characteristic appearance, and this is confirmation that the arsenic is actually substitutionally incorporating into the silicon lattice. So we are most of the way there to having substitutional electrically activated dopants. The next criteria is that we need a self-limiting saturation coverage of the precursor molecule on the silicon surface. This is so we can generate metallic interconnects using the dopants, and the concentration needs to be above the metal insulator transition and below 1 monolayer. So here we have saturation coverage imaged by the STM. What we note is that there's no ordering either long or short range in this layer. In the phosphine case, there's actually some areas of local ordering. This is because the phosphine PHX moieties are able to move around and order, but the ASHX moieties, as we know, are immobilized and do not move around, resulting in totally disordered layer. Now, if we heat this up to 350, we once again see our telltale ejected silicon, and if we count the number of silicon here, we get an estimate of the number of arsine that's incorporated into the surface. This turns out to be 24% of a monolayer. And in fact, if you alter the dose rate of the arsine, this number does not change. And this can be explained by the speed of the dissociation reaction. And here we have a kinetic Monte Carlo simulation of the dissociation. And what we find is that in all accessible partial pressures, the ASH2 concentration doesn't change. This is because the reaction's too fast. And what we end up with is within plus or minus 1% invariant coverage of total arsenic atoms on the surface. This is in contrast to phosphine, where changing the partial pressure can adjust the coverage of phosphorus atoms within about 10%. Okay, so the next criteria is area selective absorption, compatibility with the hydrogen resist. Here we start with a clean hydrogen terminated silicon surface. We pattern a 50 by 50 nanometer block, expose it to saturation arsine, and here we see our disordered arsine coverage. Heat this up to 350, and once again we get our ejected silicon, confirming we have incorporation of the arsenic only within the patterned region. Of course, we want to do this on an atomically precise scale, and here we pattern just a single dimer row of desorbed hydrogen and expose this to the arsine, and we get selective absorption just along that dimer row. And if we look close, we see some ordering within the chains there. Okay, so now on to the fifth criteria, which is that we need to be able to confine the arsenic beneath epitaxial silicon. We want very dopant patterns. Turns out if you use the recipe that works for phosphorus, which is a constant 250 degrees Celsius during overgrowth, the arsenic segregates to the surface, as is shown here in this secondary ion mass spectrometry measurement. But you can adjust the sample temperature during overgrowth and actually suppress this segregation and confine the arsenic at the doping plane. Here we do it with a low temperature throughout the entire growth, but if you use low temperature just at the start and then crank the temperature up, it still works, and we end up with confined dopants in the doping plane. We can fine-tune this and add a rapid thermal anneal just after this low temperature locking layer, which gives us good crystallinity in the overgrowth and maintains the confinement at sub 2 nanometers. Turns out if you use this locking layer technique with the phosphorus, you also get better confinement, but compared to the arsenic, it's less confined. So arsenic turns out to be the better confined of the two species once we have accounted for and controlled the segregation. So you can now imagine possibly doing some different advanced fabrication with these different responses to the substrate temperature during overgrowth. Okay, the final criteria is that we need dopant activation and ohmic contacting. And I won't go into much detail here other than to say that we can deposit aluminum contacts onto the dopant patterns, and we measure ohmic IV curves, and we can in fact tune the properties of the delta layer by adjusting this rapid thermal anneal temperature, and they give us a comparable properties to the saturation layers compared to the phosphorus. So both phosphorus and arsenic are both usable as electrically activated dopant layers in the silicon. Okay, so we've met all the criteria, and arsine and arsenic are compatible with scanning telemicroscopy, hydrogen resist lithography. So now we've introduced this new dopant, where to? Well, the idea now is to expand the materials palette further, and our group and other groups internationally are all working on this. Shortly after the arsine was introduced, a group at IBM Zurich successfully introduced diborane, and in fact built some atomically thin PN junctions out of this. And then more recently, there's some theory explaining the diborane surface chemistry coming out of Sandia National Laboratories, United States. We've actually teamed up with the group from Zurich and done some measurements on these atomically thin PN junctions using electrostatic force microscopy. And there's ongoing work looking for other precursors, including chlorides and a lot of focus on the acceptors. UNSW initially, shortly after developing the process for silicon, also did it for germanium using phosphorus and demonstrated full device fabrication. We, in our group, have recently been working on introducing arsine to germanium, and this is work led by PhD student Emily Hoffman. And so far, she's demonstrated the first few criteria in that the arsine dissociates, incorporates, and also provides a usable self-terminating saturation layer. Finally, there's work on new resists, and these are focusing on halogens. And so far, it's demonstrated you can use a chlorine resist in place of the hydrogen resist with a few important differences. Okay, so in summary, I've shown you that arsine and arsenic work with this STM fabrication procedure. Here's the paper if you want some more details. And the highlights are that the arsine is dissociating in this unique structure where the arsine is immobilized, and this results in a dose rate independent saturation coverage, and also the arsine is more confined than the phosphorus. Finally, this is work done by a number of people, both at UCL, LCN, including some former and present PhD students, and also collaborators both internationally and in the UK. Thank you.
Well, thanks very much for all three of those. Really interesting to see how, despite the fact the molecules were very different and the fields of application were very different, we were seeing some of the same techniques, the use of tips or microfabricated electrodes to apply forces to the molecules and looking at their response to those forces across those very different topics. So we have a few minutes for questions. Please stick a hand up or type something in the chat. I'm going to start with one to Binoy. Binoy, I'm fascinated to know what was happening with the cell membrane during your whole cell experiments. Were you inserting your electrode through the membrane? And if so, what happened to the membrane?
So we checked whether it is permanently damaging the cell. So what we did is we did the traditional cell viability assays using calcine and other like trip and blue, all those. So we don't really see much of a cell destruction there. So we assume that because the tip is very small, like the footprint is like in the order of few tens of nanometer or the most 100 nanometers. So it's kind of maybe just gliding through the membrane and going inside and coming out without creating a permanent rupturing of the membrane. So the cell stays intact. Very recently, we did the same assays in neurons, and we could keep the cell intact as long as 48 hours after the biopsy as of now. Yeah, so they are staying intact.
Thank you. I have a bunch of other questions, but I think Michelle applies the whole thing. Anyone else want to ask a question? Please either stick a hand up or type something in the chat. Well, maybe I will ask one more for Ismael, if I may. Ismael, you showed these plots where which you kind of averaged over a lot of experiments, right? Showing the conductivity as a function of the event duration. And I'm just intrigued to know, there's obviously a lot of information in the average of those, but I'm guessing there's probably also a lot of information in the noise and the fluctuations. Have you tried to analyze that, and have you managed to get anything out of it?
Yeah, that's a very, very, very good point. I mean, I didn't have time to go into all the details, but yes, in this actually single molecular electronics field, we have ways to analyze the noise. And from the noise analysis, you can gather some mechanistic information. So we haven't done that for proteins. The noise analysis of proteins is quite complex at the moment. So we are trying to, see if we can just, design these methods, right, to extract information. But essentially from like simple synthetic molecules, to give you an example, you can see if the noise, from the noise of the current flowing through the molecules, if the electron is going through the molecule, through bond, or for example, through gap, you can see, actually identify that kind of events. Right? So in case of a protein, it'll be very interesting because if you have pathways, or you have something like more random, right, distributed, or you have some sort of interference, right, quantum interference inside between different pathways of the electrons, that should come out in the noise analysis. So yeah, very good questions, and yes, this is an open thing that we're trying to analyze. Yes.
Okay, thank you very much, Ismael.
Thank you.
Any further questions? I know we're pretty much at the end of our time. I'm just looking at the participants list in case anyone wants to put a hand up. Taylor, I feel it's only fair I should ask you something, even though I should probably know the answer to this one. Why are those locking layers so amazingly good for the arsenic? It seemed almost like magic that little bit of growth at such a low temperature had such a beneficial effect. Do we understand why that is?
Can you?
Yes.
There I am.
Yes.
Okay.
We got you.
Yeah. I think this is. I think that once you have the arsenic fourfold coordinated, it's locked in position, and it has this low diffusivity due to its size in the lattice. But when it's exposed on the top, then it easily can exchange positions with the incoming silicon. I mean, you can explain this just energetically. Some sort of deep insight into why this happens. I'm not sure. All
right. Well, since it is 3 o'clock, I think we should thank all three speakers for doing great talks. Thank you for being clear and accessible to all of us. That was really fantastic. And like I said, that was a really interesting three really interesting views of different aspects of single molecule science in the LCN. So thanks very much, and we'll see you at the next set of seminars in a month's time. Thanks, everyone.